So the work that we do is focused on this metabolic network. I always present it in this sort of a subway map view, and we try to understand the different factors that influence this metabolic tweaking. So this involves nutritional factors, but also pharmacological factors that somehow influence this metabolic network. So can maybe activate metabolic pathways, can inhibit metabolic pathways, but also induce or sort of provide the substrate for a certain metabolic pathway, like glucose supplementation or fatty acids or anything else. We work on of course, the role of DNA. So this involves mutations of the DNA that cause inherited deficiencies of these metabolic pathways, but also transcription factors that can be activated, or specific metabolic switch points, branch points in these metabolic pathways that can be activated or inactivated to move from one metro line to the other, for instance. And in the end, what we hope to achieve is that we want to understand metabolic health. How can we improve metabolic health in the context of inherited metabolic diseases as well as age-related metabolic disease? And so the group that I lead is called Translational Metabolism. And the way we look at this and the way we see this translational aspect is essentially in two ways. On the one hand, we try to bridge the gap between inherited metabolic diseases, shown here as the newborn screening card, and we try to bridge the gap with age-related diseases. And sometimes it's pragmatically also, because the funding is usually available for age-related diseases, maybe a little bit less for inherited metabolic diseases, although fortunately that's changing a little bit. We try to bridge the gap, for instance, using molecules that are initially discovered in the context of aging and trying to use them for metabolic diseases, and at the same time also inherited metabolic diseases, sometimes also sort of reflecting in an accelerated aging phenotype. So we go back and forth. And the second way of illustrating this is going back and forth between model organisms. Our goal, of course, is to treat humans with a particular disease. But to achieve that, to get there, we still need other types of models, including mice, including C. elegans, which we particularly use for our aging work, but also other types of models, including iPS-derived models, cell models, organoids, flies, fish, whatever you can think of. So this is the translational step that we try to take. And the work that I will present today is mainly focused on NAD, nicotinamide, adenine dinucleotide, NAD+, particularly, which is a molecule that most of you, or if not all, will know from your textbooks, mainly as a cofactor in energy metabolism, as a redox cofactor, mainly in balance with NADH. But in recent years, probably the last 2 decades or so, there's more and more evidence that NAD can play a more pleiotropic role, because NAD is also the substrate for several families of enzymes, including the PARPs, the poly-ADP ribose polymerases, the sirtuins, and the C-ADP ribose syntases. And these enzymes, in turn, are not regulating just one reaction, but they are regulating many, many different reactions in a cell. And therefore, NAD as a substrate for these enzymes can play a very versatile role in managing cellular homeostasis. And by now, we estimate that NAD participates in more than 500 enzymatic reactions, covering basically all the metabolic processes in a cell. So obviously, this is an important molecule, and it's involved in many of these aspects that are shown here on the left: chromatin regulation, epigenetics, signaling processes, cell division, DNA repair, inflammation. It's regulated in a circadian fashion, so we see it oscillating throughout the day, and also obviously mitochondrial function and redox reactions, which is what it's mostly famous for, at least initially. And so maintaining this NAD homeostasis is crucial for managing cellular function. So for a molecule that has such a central role in managing metabolic function and cellular homeostasis, it seems quite logical that NAD homeostasis is maintained in a very strict sense. And to understand this a little bit, it's important to understand the synthesis and the consumption pathways that both lead to a NAD homeostasis. And this is a quite simplified version of that scheme of how NAD is synthesized and consumed. So NAD, we take it a little bit as a center. And on the one hand, NAD is being synthesized from a variety of different molecules. So tryptophan, the amino acid, is being converted in a multi-step process to NAD, but also niacin, nicotinic acid, or vitamin B3 is also converted to NAD. And also some more other precursors for NAD or NAD boosters, as I will also call them, like nicotinamide riboside, nicotinamide mononucleotide. These are all molecules that in a few steps lead to the synthesis of NAD. There's also other factors like AMPK, the energy sensor, which can also increase the synthesis of NAD indirectly. And AMPK in turn is regulated by fasting, by exercise, and resveratrol, for instance. And so each of these factors indirectly will also lead to an increase of NAD levels. At the other side of the balance, so this is the synthesis side. On the other side, we have the consumption of NAD, which also obviously lead to managing this balance of NAD levels. And so NAD is being consumed by several families of enzymes. So I already mentioned CD38 as one of the cyclic ADP ribose synthases, the PARPs and the sirtuins. And we know that regulating the consumption of these different consumers, like for instance CD38, if you inhibit the consumer, this will lead to an increase of NAD levels, which then become available for the other families of NAD consumers. So playing around with synthesis and inhibition of consumption gives us a lot of opportunities to play around with NAD and figuring out what is the importance of NAD for physiological processes. We also know, I guess it's logical if you think about the central role of NAD in metabolism, that an unbalanced NAD homeostasis leads to various diseases in humans. And this is also where our translational work really comes out, because we combine inherited metabolic diseases of NAD biology to more acquired diseases. So on the one hand, we have the primary deficiencies, or at least I call them the primary NAD deficiencies, and this is because they're genetically determined, and they are genetically determined to regulate the synthesis of NAD, so the primary synthesis of NAD. So one of them is Hartnup disease, amino acid transport, also Howe and Kinu deficiency, NMNAT deficiency, NADSIN, so really the NAD synthesis, but also the detoxification of some side products of NAD. And so this also regulates primarily NAD homeostasis. So these deficiencies have been described. I'm sure there will be more that are already described or will be described in the future. And they primarily affect NAD biology. But besides these primary deficiencies, there's also several diseases where a change in NAD occurs, but this is not the primary defect. So I call them secondary NAD deficiencies, where other factors are compromised and together lead to a reduced NAD content. And the most, well, I'm not sure it's the most famous, I shouldn't say that, but one of the historic examples of NAD deficiency is a disease called pellagra, the disease of the 4 Ds. And while saying this, I realize I shouldn't say it because I always forget what the 4 Ds stand for. Is diarrhea, dementia, and death. That's, I think, the 4th D. It's a very serious disease, at least in the past, when, and especially in countries where the diet is primarily corn-based, this strictly corn-based diet induces this vitamin deficiency, which then causes pellagra. But also other diseases where there's a secondary deficiency of NAD, glutamine synthetase deficiency, mitochondrial myopathy, but also aging and age-related diseases, where we do know from the work that I will show you in a couple of slides that NAD becomes depleted with age. If you want to know more about any of these diseases, either the primary or the secondary, I encourage you to read this review that we published last year that covers all of them in more detail. But for today, we'll focus more on aging and age-related disease as the primary focus of the work that ultimately leads us back at the end of the presentation, circles back to the inherited metabolic diseases and NAD. So first, a couple of slides on aging. So we've been interested in aging ever since I did my postdoc work on diabetes and obesity, and then realized step by step that aging and some of these common metabolic diseases are very much linked. So I don't think I have to explain this to you, and the population across the globe is really getting relatively older. So the proportion of people over 60 years is growing rapidly. I mean, judging by the colors, this happens first in the westernized countries, but across the globe, you see that this proportion of older adults is increasing quite rapidly. And this in itself is not a problem, of course, that's totally fine. But the problem is that we do know that with aging come various diseases that become more prevalent. So this affects diseases of the brain, Alzheimer's, Parkinson's, heart conditions, atherosclerosis, myocardial infarctions that occur more frequently in the older population, and diseases that are associated with, for instance, fat and muscle tissue, like, for instance, diabetes and obesity. So we do know that aging is a major contributor to these various diseases, and we know that there's pathways that we can act on to reduce the burden of disease. And this is what we've been primarily focused on ever since this paper that we published in 2010, realizing that the pathways that we were studying in the context of diabetes and obesity, for instance, the sirtuins, as major drivers of mitochondrial function, but also insulin receptor, the mTOR pathway, that these were also the pathways that are associated with longevity and lifespan regulation. So we know that, for instance, the caloric restriction pathway, which is associated with activation of sirtuins and mitochondrial function, is a pathway that, when activated, extends lifespan. And on the other hand, the caloric excess pathway, so the more anabolic pathways like the insulin IGF pathway or the mTOR pathway, they are actually associated with a shorter lifespan, and you need to inhibit any of these key players, the ones that are highlighted here in orange. Inhibition of any of these players will extend the lifespan, so rather than activation. So we realized that quite a lot of these pathways overlap between the disease context that we were initially interested in, obesity, diabetes, and that there's a lot of overlap with aging. And so we started digging deeper into which mechanisms and which key players in cellular and molecular biology underpin these changes in aging. And one of the molecules we focused on was NAD, especially because NAD is the key substrate for the sirtuin pathway, which was the main regulator that I was focusing on at the time. So we measured NAD in mice. This is shown here for mice in the liver of young mice, which is about 3 months old, and compared that to mice that were 2 years old, which is, for a mouse, I don't know how familiar you all are with mice, but 2 years is pretty old for a mouse. And we do see that in the liver of these mice, but also in other tissues like the muscle, we see that NAD declines with age. So this was a first indication that actually there is a change in NAD with age. And this is associated with the activity of PARP. I don't say this is causing it, but the PARP, as you can see here in the Western blot, is pyrrolation activity, autopyrrolation of PARP. We do see that there's a strong signal in the old animals showing that PARP is being activated with age. And so the idea would be that PARP activity would then consume a lot of NAD, and therefore NAD goes down. We haven't causally demonstrated that, but that's the general hypothesis. And so we've seen this in mice. We've also seen it in C. elegans, in worms that are older. We also see that NAD goes down. But the main question was at the time, more recently actually, is this also true in humans? And this has been, well, not been studied for a very long time, but we've started studying this a few years ago in collaboration with Lotte Grevendonck, Joris Hoeks, and Patrick Schrouwen at Maastricht University Medical Center, also in the Netherlands. And this is a study that was spearheaded by George Janssens, a PI in my group. And what we did is basically we took biopsies, especially the team in Maastricht is actually the one doing the clinical studies, took biopsies, muscle biopsies from younger individuals, so typically students, and from older adults who were between 65 and 80 years old, and they were subsetted according to their physical activity levels. So we had trained individuals, I would really call them strong athletes. I mean, they take about 14,000 steps a day, which is amazing. We have a group that we decided to call normal older adults, but they take still 10,000 steps a day. So that's still, for me, at least in the type of job that I have, is still a lot, 10,000. And we had a group of older adults that were physically impaired. So we really had evidence that there was some impairment in their physical function. They still take over 6,000 steps a day, but they were functionally impaired. So we were able to get the muscle biopsies, even from the impaired group, which was a bit of a challenge because they already have impaired muscle function. They still allowed us to take a muscle biopsy. We took the muscle biopsies and we performed metabolomics on these muscle biopsies to identify metabolic changes that can maybe explain the difference between younger individuals and older individuals, and maybe also the difference between trained older adults and non-trained older adults. And so this is the data that we got. On the left, you see the volcano plot with all the metabolites annotated that are significantly changed between normal older adults versus young. So we see quite a few changes. They are also highlighted here in the heat map, where the red signal means it's high and the blue signal means it's low. And the one that stood out for us, also because we are, of course, from an NAD background, is this NAD molecule, which is high or relatively high in the younger individuals, and then very much so reduced in older individuals. There are several others that are also very interesting related to the NAD pathway, like quinurinic acid. Also ophthalmic acid is an interesting one because it has to do with the reactive oxygen species, although it's also a little bit different than really reactive oxygen species. So we're looking into several of these changes still today. But the one that stands out, like I mentioned, is NAD, which also seems to go down with age. And indeed, if you look at the individual values and you also, again, classify by physical activity level, you can see that there's this stepwise approach where older adults have a lower, generally lower abundance of NAD in the muscle, but this is relatively well preserved or maintained in athletic elderly, and it's the lowest really in older adults with an impaired muscle function, suggesting, but not causally demonstrating, that physical activity will improve your NAD levels. That's, of course, I mean, the causal demonstration of that fact is something still to be established. So we do see in preclinical models, mice, C. elegans, and now also in a clinical model, so in a clinical setting in humans, we see that NAD goes down with age. And the question is, can we fix this? Can we fix this age-related decline in NAD? And does that improve age-related diseases? I will show you several studies, several examples of studies where NAD depletion has been corrected with various NAD precursors. So this is the first example. I already mentioned at the start, I touched upon this pellagra as a disease that is basically the first description of an NAD depletion syndrome. This is dog body weight. This is dogs with what they call black tongue disease, which is the same as pellagra. And these dogs, they have a very strong decrease in body weight until nicotinic acid is being given to these dogs. And all of a sudden, because nicotinic acid is a precursor of NAD, these dogs gain weight quite rapidly until the vitamin is again removed and it sort of stabilizes. And so the authors at the time, it's some years ago, described that the single dose of this specific nicotinic acid gave a phenomenal response in dogs suffering from black tongue disease. Appetite improved immediately, animals showed a growth response, diarrhea disappeared, and growth continued uninterruptedly. So this was really a very striking example of a very early on treatment of a metabolic disease, metabolic deficiency with a vitamin that then improved the pathophysiology. So I mentioned nicotinic acid is one of them, but we have several precursors, several NAD boosters by now that we can employ to improve or restore NAD homeostasis. So one is tryptophan as an amino acid. We always considered tryptophan one of the minor contributors to NAD biology. The major ones are nicotinic acid, nicotinamide, which is both a precursor and a downstream product of the NAD consumers. Nicotinamide riboside, which is a more recent addition to this portfolio, and also nicotinamide mononucleotide, which also is a relatively recent. Both of these molecules, they sort of exist or have been studied only for the past 10 years in preclinical models and only for the last, let's say, 5 years in clinical models. So we know that each of these molecules, nicotinamide riboside, NMN, and several of the others, in a few steps. So here we have nicotinamide riboside, we have nicotinamide mononucleotide. In a few steps, they will be converted to NAD and elevate the NAD levels intracellularly. So what has been published on these precursors? So this is a study that we did ourselves in mice with obesity or diet-induced obesity and diet-induced insulin resistance. You can see here the white one, the white circles are the animals on a high-fat diet. These ones are the animals on a low-fat diet. And you see that on the high-fat diet, they gain significant body weight. But if you then supplement the high-fat diet with nicotinamide riboside, they gain less weight. And there are several improvements. I will not go into all of them, but this is, for instance, in the brown adipose tissue, where the mitochondrial density after supplementation with nicotinamide riboside is much more pronounced compared to the vehicle-treated animals. And so we see after nicotinamide riboside treatment in this preclinical model, this mouse model, we see a massive improvement in insulin resistance or insulin sensitivity. And these mice are gaining less weight. This is a second example of a study we did ourselves also where we treated C. elegans, so worms, with nicotinamide riboside. This is a so-called lifespan curve where at the start of the experiment, all the worms are still alive. And then if we follow the trajectory of the red line, which is the control worms, we see that after roughly 4 weeks, all the worms are dead. We get to the 0, maybe 5 weeks. But if we supplement these worms with nicotinamide riboside, we do see that the median lifespan is very significantly extended, extending lifespan. And we tested the mechanisms that are involved. So this is a protective pathway called the mitochondrial unfolded protein response, which is activated. This is the dark blue line bar, activated after nicotinamide riboside exposure, but also the defense to reactive oxygen species, which was also improved after exposure to NR, all contributing to this extended lifespan. And then there's some work also that was done in the former lab that I did my postdoc, but I was not involved in any of these studies, where they showed that, for instance, the proportion of muscle stem cells improves after exposing animals to nicotinamide riboside. And this is both in young individuals, young mice, as well as in old mice. And even if you start treating mice after 2 years of age, you still see an extension of the lifespan after exposure to nicotinamide riboside compared to a chow diet, sort of control diet, where you don't supplement. So even late stage supplementation of this vitamin B3 homologue still extends the lifespan. And this is the final example that I wanted to show. This is again C. elegans, but also mice, C. elegans on the left, mice on the right. This is a model for neurodegeneration, where these worms are genetically engineered to undergo this neurodegenerative phenotype. And you see that within a few days, these worms become paralyzed. So this is the mobility that we measure. But if we supplement the worms with nicotinamide riboside, the orange line, you see that the paralysis is less pronounced compared to the control. And the same is true in mice. So this is a mouse model where the time for freezing indicates a sort of a memory function in these mice. And so in the wild type, there's about 60% of the time freezing. It's a fear conditioning assay. And if you then do this neurodegenerative model, they actually spend less time freezing because they don't remember that they should freeze. But if you then treat these mice with nicotinamide riboside, all of a sudden, it brings it back to the control scenario. Again, underscoring how these NAD precursors can improve various parameters of age-related disease. So we've seen by now that in all these models, preclinical models of age-related diseases, that NAD boosters, NAD precursors improve function. So it could be liver function. There's studies published on fertility, insulin secretion, basically any and all of these various parameters where NAD takes center stage and is improving the phenotype. But how about humans? So this is a study, there are several studies have been published. This is a study that we've been involved in where nicotinamide riboside was given to relatively healthy individuals, but they were sort of pre-diabetic. And nicotinamide riboside was given for a period of 6 weeks. There was a washout in between, and then they were switched either to the, so either they start placebo and then go to nicotinamide riboside or the other way. And from these individuals, we did a detailed clinical phenotyping for their glucose homeostasis, or their glucose handling, as well as a molecular phenotyping and biochemical phenotyping in muscle biopsies that were taken. And so the first thing that stands out, if you look at the muscle of these individuals on the nicotinamide riboside supplementation, we do see that there is an increase in various, so we do metabolomics on these muscle samples, we do see that there's an increase in several of the metabolites of NAD. So this nicotinic acid adenine dinucleotide, which is sort of a precursor, as well as methyl nicotinamide, which is a downstream product. We do see that they are increased in abundance. At the same time, we don't see any change in NAD whatsoever in the muscle of people undergoing this supplementation. And we don't really know what's going on. It seems that NAD is maintained at a stable level. It seems that there is, at least based on these data, it seems that there is flux through the pathway because we see these downstream products being increased. So we think something happens there. It's taken up, it's being used. And so that's why we do see the downstream products being elevated. But NAD itself is being fairly steady. And that's what we also see in other trials that are performed by other groups. Still, I mean, so we do think that NAD goes in. We do think that there is metabolism of NAD in these individuals that are taking it. But we do see, if we look at insulin sensitivity, substrate, these kind of parameters of glucose homeostasis, whole body glucose homeostasis, we don't see any changes, significant changes in the rate of appearance or disappearance of the glucose in a clamp, for instance. So there is no evidence so far, at least from our clinical trials, that nicotinamide riboside is doing much. So how come? Why is there limited translatability from these mouse studies, these preclinical studies that are spectacularly positive to the human situation? So the question is why this happens. We think that maybe the duration of the studies may be simply too short. Mouse studies, we typically do 8 to 12 weeks. Human studies are also maybe 6 to 10 weeks. They're not very long. Maybe the dose is not right. Maybe it's also the focus on so far, at least for most of the trials, on people with a relatively healthy life. They are pre-diabetic, for instance, in the study that we did, but they're not full-blown diabetic, very sick, etc. So they're relatively healthy, I would say. So that could also be the reason. Anyway, we need to find other ways to improve this NAD balance. And so we decided to focus also on molecules that maybe have a more pronounced effect on the intracellular NAD pool, hoping that this will translate also to a better NAD situation in people with disease. And so this is when a postdoc approach, or he was a PhD student at the time, and joined me, our lab as a postdoc, is Ruben Zapata Perez. He now leads his own group at the University of Murcia in Spain. And he came with this procedure. He's an enzymologist, biochemist by training. He came up with this procedure of using NADH as a molecule and then incubating that with a pyrophosphatase from E. coli, generating NMNH, which is another NAD precursor. And then he has this fancy purification technique. I keep calling it just fancy purification, just for the sake, because I don't understand really what he's doing. But he has this fancy purification technique that allows him to isolate this pure NMNH. And the question was, can we use this NMNH as a precursor for NAD? And so the difference is really this H. So we knew NMN, nicotinamide mononucleotide, as a precursor for NAD. We already knew it for about 10 years. And now NMNH is a new one, new kid on the block. And can that one also increase NAD? And so this was basically the first graph that he showed me after going into the lab, exposing different cell types. AML12, a liver cell line. This is a adipose tissue cell line. Again, a liver cell line, fibroblasts, a neuroblastoma cell line, and HeLa cells. He exposed each of these cells to either NMN, so I would say the old-fashioned NAD precursor, as well as NMNH. I don't know why we have this old abbreviation. It was a secret abbreviation at the time. It is NMNH. So he exposed them to NMNH, and in each of these cell lines, I mean, 10 years ago, we thought this was spectacular, like a doubling in NAD levels in a cell. Now, all of a sudden, we had a 10-fold increase of NAD in each of these cell lines, except maybe the HepG2, which are a bit less responsive. But NMNH is very potent in inducing NAD levels in all these different cell lines. So I won't go into all the details of the study. It has been published, but what Ruben did is to look at the enzymology of these pathways, look at the different enzymes that are involved in converting NMN, the classical precursor, into NAD, and comparing each of these steps between NMN and NMNH. And he found that the majority of the pathway is identical. So there is still CD73, which is identical. It's still the ENTs doing the transport. It's still the NMNATs that catalyze the last step of the synthesis. But the difference is mainly here. The difference for NMN, the classical one, is converted to nicotinamide riboside, and then it's the nicotinamide riboside kinases that convert it back to NMN intracellularly. But for NRH, or for NMNH, it's converted to NRH, which is then integrated or imported into the cell. And then it's the adenosine kinase, which actually catalyzes this following step. So there is a difference at the level of this phosphorylation step, which seems to be the main driver of this differential response between NMN and NMNH. So the next question obviously was, does it also work in vivo? So in cells, of course, that's very nice, but does it also work in vivo? Is NMN a potent NAD precursor there? So the study that we did is actually a very simple study. So we only injected twice in the mouse, and we compared to NMN, 250 milligram per kilogram per day, which is on the lower side of what people typically use for NMN. We compared that to NMNH. We inject intraperitoneally, and then 20 hours later, we inject again, and then 4 hours later, we sacrifice the mice. Throughout the study, at different time points after the first injection, we take blood samples from the tail vein, and then after 24 hours, so 4 hours after the second injection, we take all the tissues and we follow up on the biochemistry there. So this is actually what we saw when we did this time course experiment. So the first injection takes place here at time 0, and 1 hour later, we measure NAD in the blood, and we do see that NMNH, not only in cells, as I showed before, but also in vivo in a mouse, is much more potent than NMN. And even after 4 hours, and even after 20 hours, so this blood sample was taken right before the second injection, even after 20 hours, we still see a high level of NAD in mice that are injected with NMNH rather than NMN, because that one goes back to the baseline. And this is still high after the second injection. So what happens in the tissues? Same as with the cells, especially in liver cells, we see that NMNH is very potent. So we see with NMN, there's an increase in NAD levels, which again, we were always very positive about. So this is a doubling of NAD levels in the liver. But with NMNH, it's much more potent. It's a 4-time increase in NAD levels. And even in tissues that are notoriously more difficult for improving or elevating the NAD levels, like gastrocnemius muscle, also the brain, which typically don't respond very well to NMN, they do respond, although not spectacularly, but they do respond to NMNH. Still, the majority of the NAD increase we do observe in liver and kidney, which are always the most responsive tissues for these kind of treatments. I promised you I would circle back to inherited metabolic diseases, and this is so we did find these precursors, and we do see in preclinical models of age-related disease that these precursors for NAD do have a significant, do result in a significant improvement of physiological and pathophysiological processes. And we were studying that still, and I saw a presentation from Dina Damschroeder, who was in the lab of Robert Wessels at Wayne State University, where they studied a fly model, a Drosophila model for Barr's syndrome. And this triggered me because Barr's syndrome is my first scientific love, basically. It's the topic of my PhD some time ago. It's a rare disease, probably affects 250 individuals worldwide, at least that we know of. In the Netherlands, we have about 10. And it's a multi-system disorder that is characterized by cardiomyopathy, skeletal myopathy, so quite a number of these individuals become wheelchair bound, and also neutropenia they suffer from, which can be cyclic neutropenia or not. Not all the patients suffer from it, but it can be really compromising quality of life for these individuals, and it's caused by mutations in a gene called tafazin. I can tell you a whole background why it's called tafazin, but I'll skip it. I'll skip that for now. And actually, our lab was involved in the initial discovery that cardiolipin, a lipid of the mitochondrial membrane, is the one that is defective in cells of individuals with Barr syndrome. And also, this causes a massive mitochondrial dysfunction because cardiolipin, the lipid, also acts as sort of a glue in the inner mitochondrial membrane, sort of holding the respiratory chain complexes together for efficient electron transfer. So this is a disease that we've been interested in for a long, long time, also because Professor Barth, who first extensively described the disease, was actually from our institute. So this is a sort of a historic research line that we still actively pursue to this day. And so what Dina and Robert did in Wayne State University is to work with these flies. They typically sit in these tubes, and they tend to crawl up in the tube. And what they devised is this machine called the power tower, and essentially it bangs the flies back down in the tube. And so every time the machine rotates, the flies go back down in the tube and they crawl back up. And what you can measure is the time to exhaustion of the flies. And this is shown here. I will first go to the basics. So the wild type strain is the black line. And you can see that at the start of the experiment, all the flies are still running around. Switch on the laser pointer again. At the start of the experiment, all the flies are running. They go back up in the tube. And then slowly they become tired. And after 300 minutes, roughly 350 minutes, all the wild type flies are exhausted. The flies with Barr syndrome, the tafazin mutation, we saw that the flies become already fatigued after 150 to 200 minutes. So there's really a 50% reduction in their exercise capacity. But if you then supplement these flies with nicotinamide riboside, the NAD precursor, all of a sudden the flies with Barr syndrome all of a sudden have a similar kind of exercise capacity as the wild type flies, also on nicotinamide riboside. We do see that in the wild type also it improves a little bit, but the improvement in the flies with Barr syndrome is really spectacular. And this opens up a lot of opportunities also for treatment. I think it's really interesting to look in this direction. So what can we do with treatment? There's a lot of pros and cons. Well, there's a lot of discussion, at least, on how to approach such a clinical study. But we were also helped here by another study of a close collaborator of mine, Eija Pirinen, and in the lab of Anu Suomalainen in Helsinki, where they tested niacin, nicotinic acid, also a precursor for NAD. They tested it in the context of mitochondrial myopathy, so depletion or deletion of mitochondrial DNA. And so they tested niacin, and after 4 to 10 months, typically after 10 months, the effect is more pronounced. They see that the exercise capacity, so in this case, 6-minute walk test, is significantly improved in the patients. And also this is a muscle strength, and this is the back muscle, but they tested several muscles. The muscle strength also is improved after 4 to 10 months on niacin. So overall, they've seen that niacin treatment in patients with mitochondrial myopathy improves their NAD, it improves their muscle strength, it increases mitochondrial mass, increases OXPHOS function, exercise capacity, it maintains sort of a nice glycogen level, it reduces liver fat, reduces lactate, and reduces hemoglobin. And so altogether, these various parameters are very, very much so indicative of an improved disease phenotype, and for us are a very interesting basis to look for opportunities to start a clinical study also in the future for Barr syndrome. At the same time also, for instance, still doing the mouse work, the mouse studies, to identify the mechanism by which NAD improvement also improves the clinical phenotype. So that brings me to the conclusions of my talk. We do see that NAD declines in tissues across various organisms, mice, C. elegans, but also humans. We do see that this associates with health status. So people who maintain an active life tend to have higher NAD levels. Although again, the causal relation will have to be established. We see that what I call classical NAD precursors, they're not so classical because they're only around for just over 10 years, but they have shown limited translatability to humans. The clinical trials, we do see some effect in one trial, some in others, but not very positive, like the preclinical models, which are massively positive, we do see that these reduced NAD precursors like NMNH, but we also identified together with a group in Switzerland, NRH, which is very similar actually. They may offer attractive opportunities to improve NAD homeostasis, but obviously there's more work to be done to apply this in a clinical setting. No clinical trials have been done with any of these molecules, and they're very unstable also. They will not pass the stomach. So there really needs to be a solution to, for instance, encapsulate the molecule to be able to use them. And I think with the data that we have for Bar syndrome in this preclinical model, in this fly model, but also the trial that was done in mitochondrial myopathy, for mitochondrial myopathy, I think boosting NAD levels could serve as a therapy, at least for some inherited mitochondrial disorders. In the brief meeting we had prior to my seminar, we already discussed a little bit about it. We, for instance, don't see any evidence of a beneficial effect of these molecules in the context of fatty acid oxidation deficiencies, mitochondrial fatty acid oxidation deficiencies. So it's definitely not a magic bullet that will treat all inherited mitochondrial disorders. And we really need to invest in understanding the mechanism to understand also which disorders it could be beneficial for. So with that, I'd like to thank the people who were involved in this work, mainly Ruben Zapata Perez and George Janssens, who were involved on our side in spearheading these projects. Several other people in the lab, including my mentors, Fred Voss and Ron Wanders. Also our pathology department was instrumental in some of the work on the kidney. University of Murcia for identifying initially these molecules as NMNH. Our clinical trial collaborators in Maastricht University, especially Patrick Schrouwen, with whom we have a long-standing collaboration now. And also at the Nestle Institute for working together with us on these reduced precursors, and Wayne State University, Dina and Robert, for the work on the flies with Bar syndrome. And of course, the funding agencies that made all of this possible, and without whom we couldn't have done this. And then I'll take the final opportunity to highlight our new project. If you happen to be a aspiring PhD student, or if you know a master student or a recently graduated master student who wants to do a PhD in the field of NAD, please make them aware that we're starting this. We have several open positions. We're recruiting 10 PhDs across these European centers. 2 will be based in Amsterdam with us. The positions are on this website there. The position is still open for another 5 days. Then we close it and we select. So if you happen to know someone, they have to be fast. Yeah, feel free to advertise that. And with that, I'm happy to stop and take any questions that you may have, any points you want to discuss.